Alright, so here are a couple of other problems. This is Pear's Chapter 3, problems 9 and 10. They're both pretty related, so I do both of them. So independent assortment is, and segregation of alleles are really the two things going on with Mendel. So Mendelian genetics deals with how genes are inherited. And so typically we'll have a couple of genes represented. Here's just one gene with two alleles. Big A and little a are alleles. So segregation of alleles means, surprisingly enough, that the alleles segregate away from each other. And they segregate away from each other in the formation of gametes. So gametes are really important. So gametes are the basis of reproduction. And, so let's say this is a cell. This is a male, those would be a couple of sperm cells. That's what we mean by gametes. And that what we mean is that big A and little a segregate into separate gametes. You can't get both big A and little a in the same gamete. You have to get big A and little a into separate gametes. Now what independent assortment means is if you're dealing with more than one gene. So here are two genes. If you get a big A, you can also get a big B. If you get a little a from here, you can also get a big B from there. And we can get two other kinds of gametes. If we get a big A from here, we can get a little b from here. That's another gamete. If we can get a little a from here, we can get a little b from here. So independent assortment is when we're dealing with two genes, saying that how these things sort into gametes. So big A and little a have to go apart. So you can get either big A or little a. So that's segregation of alleles. If you get a big A, you can get either big B or little b. Big A, big B, big A, little b. That's independent assortment. Little a, you can get big B. Little a, you can get little a, big B, little a, little b. That is what we mean by independent assortment. We'll come back to when this happens. This all happens during meiosis. No segregation of alleles or independent assortment in mitosis. Alright, so I'm going to get rid of this layer. Add a new layer. So the way we see that in a cross is we'll have big A, big B, big A, little a, big B, little b. And if we have independent assortment, we can get gametes. We can get big A, big B. We get big A, little b. We can get little a, big B, little a, little b. Now we can get that if we have independent assortment. And that happens during meiosis. So we'll come back to that. I'll show you how that happens in meiosis in a minute. But the other thing that's important is that these are all equally likely. They're all gametes. Gametes are actually cells. That's why we draw these little circles. So these are sperm cells or egg cells or pollen cells if you're dealing with plants. And they're equally likely. So we can get them in a 1 to 1 to 1 to 1 ratio. All right, so that's what independent assortment means. If you have a dihybrid, wow, that's crazy, but that says dihybrid. So dihybrid, you're always going to get these kinds of gametes. Alright, so when does this happen during meiosis? So when does it happen? Not during mitosis, only during meiosis. Meiosis is really the important thing when it comes to genetics. So when this happens is during sort of metaphase and anaphase of meiosis 1. So you have meiosis 1 and meiosis 2. And so we're going to look at metaphase 1. So there's two divisions in meiosis, meiosis 1 and meiosis 2. They each go through prophase, metaphase, anaphase, that sort of thing. The only thing we really care about is meiosis 1 and particularly metaphase. So that means we've got to put these genes on chromosomes. And that kind of looks like a K, but that's a chromosome. So that's a chromosome with a centromere, big A's there. And here's the big thing. Get ready. Another chromosome. This one looks more like an X maybe. Put a little A on there. Metaphase 1, homologous chromosomes. Line up in pairs. So then when they divide, when the cell divides, I don't know why that one keeps looking like a K, that one looks like an X sort of. Now you get these chromosomes separate away from each other. They segregate during metaphase 1 of meiosis. So they line up in metaphase, and then they'll separate during anaphase, telophase, cytokinesis, all that stuff. But it's how they line up here in metaphase 1 that's important. That's the segregation of alleles. Alright, so that's that, metaphase 1. So anytime we're talking about anything about meiosis, or anything about segregation of alleles or Mendelian genetics, it's always during metaphase 1 of meiosis. So these are alleles, big A and little a are alleles. These are homologous chromosomes. Let's see. Alright, so let's delete that layer. We'll do another one. And now we'll do independent assortment. So now that means we have 2 genes. And you could have 3 genes, 4 genes, 5 genes, 1,000 genes. But we're talking about how genes assort independently. And that means that they're on different chromosomes. So we're going to have our A chromosome. So here's a pair of homologous chromosomes. And this is a homologous chromosome that has the A locus. So locus. Loci. So a locus is the position where this gene exists on the chromosome. And this chromosome has big A alleles. This chromosome has little a alleles. These are sister chromatids. And sister chromatids are always identical. So when you have this chromosome, you have to have a little a here and a little a here. And at this one, you have to have a big A here and a big A here. So these guys are lining up just like before as a pair, homologous chromosomes in a pair, and they're going to segregate away from each other. I'm going to redraw that because I need a little more room. So I'm just going to redraw this so that this homologous chromosome, and we know it's homologous because it has the same letter gene on it. So this is the chromosome that has the A gene. This has the big A allele, this has a little a allele. So this guy is heterozygous for big A, little a. But we're also going to look at independent assortment, so we need to put another gene on there. So this guy is going to be also heterozygous for the B gene. And the B alleles are going to be on homologous chromosomes, and they're going to line up in a pair. Now, as we go through meiosis, these things will segregate away from each other. So these guys are going to separate. So there's going to be a couple of divisions so that we end up with a big A and a big B together. So this big A and this big B will go together into one gamete. This little a and little b will go together in another gamete. But you can also line up in a different way. This is what independent assortment means. Make it a little bit longer so I can fit my A on there. So we're going to have big A and a big A. Centromere, sister chromatids, little a, little a. Big A and little a always line up in pairs. They always segregate away from each other into separate gametes. But the way that the B chromosome lines up relative to the A chromosome is independent. So just because there's a big A on this side doesn't mean we have to have a big B on this side. We can have a little b on that side and a big B on this side. Now big A and little b are going to go together. And little a and little b and big B are going to go together. These are your gametes. Each of these things is equally likely. And that's independent assortment. Independent assortment means that they can line up like this, or they can line up like this. If they line up like this, they give us these guys. If they line up like this, they give us these guys. And how these things line up is random. So big A and little b can go together, big A and big B can go together, that's random. Now now in any given cell, it has to line up one way or the other. But if you look at lots of meioses, so if these are gametes that are giving rise to sperm, you get millions of sperm. And then on average, 1/4 of those sperm will have this genotype, 1/4 will have this genotype, 1/4 will have this, and 1/4 will have this. And so that's independent assortment. How's it related to segregation? It's kind of hard to say in one simple phrase, but alleles have to segregate away from each other. So alleles of the same gene segregate away from each other. That's segregation of alleles. So segregation of alleles. But different genes, so these are alleles because big A and little a are two different versions of the same gene. These are alleles, big B and little b are two different versions of the same gene, but these are different genes. So different genes, as long as they're on different chromosomes, can sort independently.